Imagine for a moment this incredible unseen war happening inside your body right now. From a tiny virus like nanometers across to maybe even a meter-long parasitic worm. It sounds wild, but your body is constantly defending itself.
Absolutely. And most of the time, you're completely unaware of this intricate network that's protecting you 24/7.
Or think about the story of the smallpox vaccine. It's almost legendary, isn't it? This medical breakthrough that literally wiped a terrifying disease off the planet through, well, sheer global effort.
It's astonishing. And these stories, they're not just interesting anecdotes. They're really our way into understanding one of the most complex and, frankly, vital systems in your body, the immune system.
Exactly. So today we're doing an essential deep dive into this amazing internal defense force.
That's right. Our mission, really, for you listening, is to provide a comprehensive, almost professional-level grasp of immunology. We want to cut through all that dense biological complexity.
Yeah, it could be dense.
And reveal the core principles, the historical milestones, and the cutting-edge insights. Basically, the kind of understanding that top professionals in the field would have. Think of this as your shortcut to really mastering that foundational knowledge.
I like that, a shortcut to mastery.
Right.
So by the end of this deep dive, you should expect quite a few aha moments. We'll aim for really clear explanations of some pretty complex biology, and hopefully give you a fresh perspective on how your own body protects itself. We'll connect these, sometimes abstract ideas directly to real-world medicine and public health issues that affect all of us.
So let's dive in. Where should we start? Maybe right at the beginning, with the history.
Perfect. Let's unpack this incredible story, starting, with its ancient roots. Okay, so let's start with the word itself, immunity. Fundamentally, it just means a state of being protected from infectious disease.
Right, and the word comes from Latin, immunis, which means exempt.
Exempt.
Yeah.
Makes sense. But while the word might be ancient, the scientific understanding of how it works, that's much more recent, isn't it?
Oh, absolutely. But the observation of immunity, this idea that a body could be exempt from getting sick again, that goes way, way back.
It's fascinating, really, how early civilizations kind of noticed this pattern without knowing anything about microbes or anything.
It really is. One of the earliest written accounts we have is from Thucydides, the historian, during the Peloponnesian War. This was back in 430 BC.
Wow.
During a terrible plague in Athens, he wrote down something quite profound. He observed that only people who had already recovered from the plague could safely nurse the sick ones without getting it again.
That's incredibly observant for the time.
It's a remarkably sharp insight, even though, it wasn't scientific in our sense. It was this early recognition that the body could somehow learn from being sick and gain protection for the future.
And it's mind-blowing when you think, like you said, the whole idea of germs, of invisible things causing disease, that was still thousands of years away.
Exactly. Germ theory was nowhere on the horizon.
And it took, almost 2,000 years after Thucydides for that idea of acquired immunity to really start influencing actual medical practices, leading eventually to vaccination.
That's right. The earliest deliberate attempts we know of to actually induce immunity were probably in the 15th century in China and Turkey. They were dealing with smallpox.
Which was just horrific, wasn't it? I mean, the stats are grim. About 30% fatal.
And even if you survived, the disfigurement could be terrible.
Yeah. So what did they do back then?
They used a technique called variolation. It involved getting people exposed to smallpox material in a controlled way, either inhaling dried crusts from the pustules or inserting the material into small cuts in the skin. The idea was to cause a milder infection to prevent the full-blown, often fatal disease. It was definitely risky, but often seen as better than catching smallpox naturally.
I guess so. So how did this idea spread?
Well, a key figure was Lady Mary Wortley Montagu. In 1718, she was in Constantinople with her husband, the British ambassador. She saw variolation being used effectively there.
And she was convinced.
Apparently so. She showed incredible courage, actually. She had it done on her own children and then championed the practice back in Europe. A very brave move for the time.
Absolutely. But the real game changer, the person everyone associates with vaccination, is Edward Jenner.
Right. Yes, Jenner, an English physician. His big discovery came in 1798. He noticed this common observation among milkmaids.
Ah, yes, the milkmaid story.
Right. Those who caught cowpox, which was a pretty mild disease, seemed to be immune to the much nastier smallpox.
So he connected the dots.
He did. He thought, well, maybe exposing someone to cowpox could protect them from smallpox. So he ran an experiment. He took fluid from a cowpox pustule
Right.
and inoculated an 8-year-old boy with it. Later, and this is the part that raises modern ethical eyebrows, of course, he deliberately exposed the boy to smallpox.
Yeah.
And the boy didn't get sick. He was protected. It was a monumental breakthrough.
Incredible. Even with the ethical questions, you can't deny the impact.
No, it was huge. Jenner's technique spread, but it took quite a while, almost another century, for the principle to be applied to other diseases. And that brings us to Louis Pasteur.
Pasteur, another giant name. His work seems like a mix of careful science and happy accidents.
Definitely some serendipity involved.
Yeah.
He was working with fowl cholera, the bacteria that causes it. He'd grow it in culture, inject chickens, they'd get cholera, die. Standard stuff to confirm virulence.
Okay.
But one time, after coming back from a summer holiday, he used an old culture to inject some chickens. And these chickens, they got sick, but they actually recovered.
Interesting. A weakened batch.
That's what he figured out. But here's the clever part. He needed more chickens for his next experiment with a fresh, virulent batch, but he was short on supply. So he used to mix some new chickens and some of the ones that had recovered after getting the old culture.
And what happened?
The chickens that had received the old, weakened culture were totally fine, protected. The new, unexposed chickens, they all died from the virulent cholera.
Wow. So the old culture acted like a primitive vaccine.
Exactly. Pasteur realized the aging process must have weakened or attenuated the bacteria, but it could still provide immunity. And it was Pasteur who coined the term vaccine, specifically to honor Jenner's work with cowpox vacca, being Latin for cow.
That's a nice touch, honoring Jenner like that.
He then went on to prove this principle of attenuation worked for other diseases too. His anthrax experiment in 1881 is a classic.
The sheep experiment.
That's the one. He vaccinated one group of sheep with heat-attenuated anthrax. Then he challenged both the vaccinated group and an unvaccinated group with deadly, virulent anthrax.
And the results were clear.
Crystal clear. All the vaccinated sheep survived. All the unvaccinated sheep died. It absolutely solidified the concept of vaccination using attenuated pathogens.
And he didn't stop there, did he? He tackled rabies next.
Yes, his first human vaccine administration was in 1885. A young boy, Joseph Meister, had been badly bitten by a rabid dog. Pasteur gave him a series of injections with progressively less attenuated rabies virus preparations.
And it worked, because rabies is usually fatal once symptoms start.
It did work. Joseph survived. And rabies vaccine is interesting because it's one of the few that can actually work after exposure, provided the virus hasn't reached the central nervous system yet.
That's incredible. Saving that boy's life must have been huge.
It was. It led directly to the founding of the Pasteur Institute in 1887, which is still a major center for infectious disease research today.
So, fundamentally, vaccination is like giving your immune system a safe preview, right? Exposing it to a harmless version of the enemy so it can learn how to fight.
That's a perfect way to put it. It lets the immune cells develop strategies, build up their defenses, and acquire that crucial long-term protection against the real dangerous pathogen. It's like a training drill for your inner army.
And that training drill has had just a monumental impact globally.
Absolutely monumental. When you get a vaccine, your immune cells get that chance to respond, to develop, to really hone their strategy against that specific pathogen, or ones very similar to it.
And this learning process creates memory cells.
Exactly. Highly specific, incredibly long-lived memory cells. These can protect you for decades, sometimes even for life.
We've seen the results. Smallpox eradication is the ultimate example, isn't it?
It truly is. 1977, the last naturally acquired case. A disease that haunted humanity for centuries, just gone from nature thanks to worldwide vaccination, an unparalleled public health victory.
And interestingly, once it's gone, you don't need to vaccinate everyone anymore, which is good because, as you said, even the safest vaccines have some tiny risk associated with them.
Right. Which brings us to another really important concept, herd immunity.
Okay, herd immunity. Explain that.
You don't actually need every single person in a population to be immune to stop an epidemic. If enough people, a critical mass, are protected, either through vaccination or because they've recovered from the infection.
They form like a firewall.
Exactly. They act as a buffer. This dramatically reduces the spread of the infectious agent, and it indirectly protects the vulnerable individuals who can't be vaccinated. Think very young babies, people who are seriously ill, or those with compromised immune systems.
So getting vaccinated isn't just about protecting yourself, it's about protecting your community too. There's an altruistic angle.
There absolutely is. Many of us might survive something like the flu just fine, but for our vulnerable neighbors, it could be deadly. So widespread immunity benefits everyone.
But this is where it gets complicated, right? This balance between individual choice and the public good. It's a hot topic.
It certainly is. And despite the overwhelming success of vaccines, there's persistent skepticism. You hear claims that vaccines cause more harm than good, leading to calls to limit childhood vaccination programs.
Now, it's fair to say all vaccines, especially ones given to healthy people, need rigorous testing and regulation, right?
Yeah.
And people deserve clear information.
Absolutely. Public trust requires transparency and rigorous safety standards, but specific claims linking vaccines to harm have often been investigated and, frankly, debunked.
Like the thimerosal claim, the mercury-based preservative.
Right. Thimerosal was used in some vaccines for decades. Concerns were raised about a potential link to autism. It was phased out of most childhood vaccines in the U.S. around 2001 as a precaution, but crucially, autism rates continued to rise even after it was removed. That pretty strongly refuted the link.
And then there was the MMR vaccine controversy.
Yes, the study published in The Lancet in 1998 by Andrew Wakefield and colleagues. It claimed a link between the MMR vaccine and autism. This caused a huge scare.
But it didn't hold up to scrutiny.
Not at all. Nearly 2 decades of follow-up research by numerous independent groups found absolutely no evidence to support the claim. Eventually, it came out that the original data was falsified. 10 of the 13 original authors withdrew their support, and The Lancet fully retracted the paper in 2010.
Falsified data, that's serious. But the damage was done, wasn't it?
Tragically, yes. MMR vaccination rates plummeted in some areas, like parts of the UK, falling from over 90% down to around 60%.
On the consequence.
Predictably, measles and mumps cases surged. Thousands of kids were hospitalized, and several children actually died from these preventable diseases. It's a stark reminder of the real-world consequences of misinformation.
So why do these debunked ideas sometimes persist? It's confusing for people.
It is. Several factors likely play a role. One is just unfortunate timing. The number of recommended childhood vaccines has increased significantly over the decades, maybe 14 different vaccines by age 2 now, compared to fewer than half that in the early 80s.
Okay.
And the typical age for diagnosing autism and some other developmental issues is often around age 2. So parents might see their child get vaccinated and then sometime later see symptoms emerge and naturally but incorrectly link the two.
Correlation doesn't equal causation, but it's an easy trap to fall into.
Exactly. Plus, we live in an age with overwhelming amounts of information, both good and bad. It can be really hard for people to sort out reliable medical advice from inaccurate claims, especially online.
This feeds into that ethical tension again. Public health versus personal freedom.
Right. Most states require certain vaccinations for kids to attend public school, but there are usually medical exemptions for those who truly can't be vaccinated. However, about 20 states also allow personal belief or philosophical exemptions.
And does that make a difference?
The data shows a clear correlation. States with easier exemption policies tend to have lower vaccination rates and consequently higher rates of vaccine-preventable diseases like pertussis or measles.
It's like the tragedy of the commons idea, isn't it?
Hmm.
Individual choices impacting the collective protection.
Precisely. When unvaccinated people cluster geographically, it erodes herd immunity in that community. This puts everyone at higher risk, but especially those vulnerable individuals we talked about, the infants, the sick, the immunocompromised, who rely on the herd for protection.
Finding the right balance is tough.
It is. There are no easy answers. But what seems clear is that opting out of rational scientific debate based on debunked fears isn't a responsible way forward.
And going back to smallpox for a second, the eradication, while amazing, had a sort of darker side.
A sobering consequence, yeah. Because routine smallpox vaccinations stopped decades ago, most people alive today have no immunity. This makes smallpox a potential bioterrorism threat, unfortunately. That's why research continues on new, safer vaccines, mainly for military personnel or first responders who might be at higher risk.
Still, despite these complexities and challenges, the overall benefit of vaccines is just staggering.
Absolutely staggering. Think about diseases that used to routinely kill or disable children just 50, 60 years ago. Diphtheria, measles, polio, whooping cough. They're virtually gone in many parts of the world because of vaccines.
The numbers are incredible. Diphtheria cases dropping from what, over 175,000 a year pre-vaccine in the U.S. to essentially zero. Measles from 500,000 down to maybe double digits or low hundreds most years.
Exactly. And it's not just preventing death and suffering, immense as that is. Think of the healthcare costs saved, the prevention of long-term disabilities like paralysis from polio, deafness from measles, brain damage. The economic and societal benefits are almost incalculable.
And there are global efforts trying to bring these benefits to poorer countries too.
Yes, organizations like Gavi, the Global Alliance for Vaccines and Immunization, formed back in 2000. It's a public-private partnership working to boost immunization rates in developing nations and speed up access to newer vaccines.
As they made progress.
Huge progress. In their first 15 years, Gavi helped reach something like 500 million additional children and prevented an estimated 7 million deaths.
That's incredible. How do they work?
Their model is interesting. They empower the developing countries themselves to set agendas and track progress with financial backing from the World Bank, WHO, donors like the Bill and Melinda Gates Foundation, and others. The vision is for all countries to eventually afford these vaccines in dollars, not pay the price in lives
But
lost.
challenges remain.
Right.
It's not all solved.
Definitely not. Big challenges remain. Designing effective vaccines for major killers like malaria and HIV is still a huge hurdle.
Right.
And even for existing vaccines, getting them to everyone who needs them is tough. Manufacturing costs can be high. Some vaccines need careful cold storage, which is difficult in remote areas. Delivery can be complex. Millions of kids still miss out.
So we need better vaccines too. Cheaper, more stable.
Ideally, yes. Inexpensive, heat-stable vaccines that don't require needles would be a game changer for global health. And of course, the fight against misinformation continues to be a major obstacle everywhere.
So while vaccines are obviously critical, especially for serious, widespread diseases, they aren't the only tool in the box, are they?
Not at all. Prevention is key. Simple things like access to clean water, good hygiene, a nutritious diet, these go a very long way in stopping pathogens from spreading in the first place.
And some illnesses just aren't practical targets for vaccination programs, right?
Exactly. Think about the common cold or cold sores. They're usually mild, self-limiting, easily managed. Developing and deploying a vaccine is a massive, expensive undertaking, so it's typically reserved for more serious threats.
Plus, some pathogens are just really tricky to vaccinate against.
Yes, some have too many different strains or variants, making a universal vaccine difficult. Others require really complex immune responses that are hard to generate reliably with a vaccine, or they don't induce good long-term memory.
Then there are treatments like antibiotics.
Right, antibiotics were a revolution starting in the 1920s. Chemical agents specifically targeting certain bacteria. Hugely effective, but not against viruses or fungi, and unfortunately, we're now facing a serious problem with antibiotic resistance.
Bacteria evolving to beat the drugs.
Exactly. Developing new antibiotics is becoming increasingly urgent.
What about antiviral drugs?
We have some, but they're often less effective or only work against specific viruses. They don't help much against common things like influenza, for example.
Which is why we need a new flu vaccine every year, because the virus changes so fast.
Precisely. Influenza mutates rapidly, so the vaccine has to be updated annually based on predictions of which strains will circulate. It's always a bit of a race.
And a scary thought is a really bad pandemic strain emerging, like the 1918 flu that killed millions.
Yeah. With modern global travel, a highly lethal and easily transmissible strain could spread incredibly fast. Manufacturing and distributing enough vaccine in time would be a massive challenge.
So immunology isn't just about fighting infections, is it? It seems much broader now.
Oh, much broader. We're realizing the immune system is deeply interconnected with other major body systems. The endocrine system, the nervous system, metabolism. It plays crucial roles in maintaining overall health or homeostasis.
And this understanding is leading to new therapies.
Absolutely. Advances in cell and molecular biology allow us to manipulate the immune response in ways we couldn't before. We can boost it, suppress it, redirect specific immune cells.
To treat what kinds of things?
Things like autoimmune diseases, cancer, preventing transplant rejection, allergies, even other chronic conditions. We're learning how to fine-tune the immune system to treat a whole range of diseases, well beyond just infections.
And studying immunity also teaches us about basic biology.
Definitely. Research into immune mechanisms, even things like studying HIV, has yielded fundamental insights into cell biology, genetics, metabolism that have applications far beyond immunology itself.
So the immune system isn't just a defense force. It's a regulator, deeply woven into the fabric of our overall health. And we're learning how to harness its power.
That's a great way to summarize it.
Okay, so Pasteur showed vaccination worked, but you mentioned he didn't fully know how. There was a debate early on, wasn't there, about whether protection came from cells or something else?
Yes, a big debate. For decades, scientists argued, was immunity mediated by cells or by some soluble factor in the blood.
Who provided the first clues?
The first real insight into the mechanism came from Emil von Behring and Shiba Saburo Kitasato in 1890. Their experiment was groundbreaking.
What did they do?
They showed that serum, that's the liquid part of blood after it clots, so no cells from animals that had been immunized against diphtheria could actually transfer that immunity to unimmunized animals.
Just the serum. No cells needed.
Just the serum. It proved there was something soluble in the blood responsible for protection. Von Behring got the Nobel Prize for this in 1901.
So what was this soluble stuff?
Well, researchers started finding different active components in immune serum. Things that could neutralize toxins, they called those antitoxins. Others could make bacteria clump together agglutinins, or make substances precipitate out of solution precipitants.
Different names for different jobs.
Initially, yes. But then, mainly in the 1930s, a scientist named Elvin Cabot demonstrated that a single fraction of serum proteins, which they first called gamma globulin, was responsible for all these activities.
Ah, so it was one type of molecule doing different things.
Exactly. We now call that protein fraction immunoglobulin, and the active molecules within it are what we call antibodies.
Antibodies,
Yeah.
okay. And why humoral immunity?
Because these antibodies were found in the body fluids, which were historically called the humors, so immunity mediated by these fluid-borne antibodies became known as humoral immunity.
And this discovery had immediate practical uses, right? Like the diphtheria story in Alaska.
Yes, that's a perfect dramatic example. Using anti-serum serum packed with antibodies from an immunized animal, often a horse became a treatment for diseases like diphtheria and tetanus. The gnome serum, run in 1925.
The dog sled relay.
Balto.
Exactly. Balto, the lead dog on the final stretch. 20 mushers, an incredible journey covering almost 700 miles in brutal conditions, all to deliver life-saving diphtheria antibodies to stop an outbreak. It's an amazing story.
And that's an example of passive immunity.
Precisely. Passive immunity is when protection is transferred using pre-made antibodies. The recipient's own immune system doesn't have to do the work. Think snakebite antivenom or maternal antibodies passed to a baby through the placenta or breast milk.
That gives immediate protection. Yes, it's fast, but it's temporary because the recipient isn't making their own antibodies and the transferred ones eventually degrade. The antibody-producing cells aren't transferred.
Which is different from active immunity.
Right. Active immunity is what you get from being infected naturally or from vaccination. Your own immune system is stimulated, generates its own response, and crucially, creates those memory cells. That leads to long-lasting, renewable protection.
Okay, so humoral immunity is about antibodies and fluids. But what about the other side of that early debate? The cells.
Well, even before von Behring's serum discovery, Ilya Mechnikov back in 1883 had shown cells were involved. He observed certain white blood cells engulfing foreign particles, things like bacteria. He called them phagocytes.
Phagocytes eating cells.
Exactly, cells like monocytes and neutrophils. Mechnikov noticed they were more active in animals that had been immunized, so he argued that cells were the main players in immunity, not serum components.
So the debate continued. Cells versus serum.
It did. But then, in the 1940s, Merrill Chase managed to transfer immunity against tuberculosis between guinea pigs using just white blood cells. This reignited interest in the cellular side.
Getting closer to connecting the two.
Yes. Then, in the 1950s, a specific type of white blood cell, the lymphocyte, was identified as being responsible for both types of immunity, cellular and humoral.
One cell type involved in both. How?
That was clarified further by Bruce Glick's experiments with chickens. He discovered there were actually two major types of lymphocytes. One type matures in an organ called the thymus. These became T lymphocytes, or T cells. The other type matures in the bursa of Fabricius in birds, or the bone marrow in mammals. These are B lymphocytes, or B cells.
T cells and B cells, so that's how the debate was resolved.
Pretty much. It became clear that cellular immunity is primarily driven by T cells, while humoral immunity, the antibody response, comes from B cells. But critically, the two systems don't work in isolation. They are deeply interconnected and collaborate constantly for a full immune response.
Okay, so we have T cells and B cells, antibodies. But one of the biggest mysteries must have been how they achieve such incredible specificity. How does an antibody know to target, say, the measles virus and not the flu virus?
That was a huge question. And the specificity isn't just for pathogens. Around 1900, Jules Bordet showed you could generate an immune response against harmless things too, like red blood cells from a different animal species. It recognized them as non-self.
And Carl Landsteiner took this even further.
He did. His work showed that antibodies could be generated against an almost limitless range of substances, even newly synthesized chemicals that don't exist in nature. And they could distinguish between molecules that differed by just a tiny bit, like a single amino acid.
That level of precision is incredible. It suggests the system isn't just reacting broadly, it must be selecting something very specific.
Exactly. This demanded some kind of selective theory. Paul Ehrlich proposed an early version back in 1900.
Ehrlich side chain theory.
That's the one. He imagined cells having various side chain receptors on their surface, like locks. When an infectious agent, a key bound to the right receptor, it triggered the cell to make and release many more copies of that specific receptor.
And he called the key the antigen.
Yes. He coined the term antigen for any substance that triggers a specific response from these lymphocytes. Now, Ehrlich initially thought one cell could have many different types of receptors, and the antigen just selected the right one to activate.
But that part wasn't quite right.
It needed refinement. The crucial insight, developed later, was that each individual lymphocyte is already committed to one specificity. It makes many copies of just one type of receptor.
So you need a huge variety of lymphocytes, each waiting for its specific antigen.
Precisely. You need an army of cells, each with a different potential target. When the antigen comes along, it binds to the B cell with the matching receptor. That specific B cell is then selected, activated, and starts dividing, making lots of copies of itself, and also secreting that receptor in a soluble form, the antibody.
This sounds like the modern theory. Clonal selection.
Exactly. The clonal selection theory, developed from insights by Burnett, Jern, and Talmage, is the cornerstone of modern immunology.
What are its key principles?
It's beautifully simple, really. First, each B or T lymphocyte expresses many copies of a single, unique receptor specific for one antigen. Second, that specificity is determined before the cell ever encounters the antigen. It's pre-programmed. Third, when the antigen binds to that specific receptor, it activates that lymphocyte, causing it to proliferate into a clone, a population of identical daughter cells, all bearing the same receptor.
So the antigen selects the pre-existing clone and triggers its expansion.
Precisely. This applies to both B cells, which then produce antibodies for humoral immunity, and T cells. T cells use their surface T cell receptors to recognize antigens, often presented by other cells. Activation leads them to proliferate and carry out various cellular immune functions, like helping B cells or killing infected cells.
And the mystery of how each cell gets its unique receptor was solved later.
Yes, that was the amazing discovery by Susumu Tonegawa in 1976, which won him the Nobel Prize. He figured out the molecular mechanism how lymphocytes literally cut and paste segments of their DNA to create unique genes for their B cell receptors or T cell receptors. It's genetic recombination happening in individual immune cells.
Wow. So to boil it down, our bodies generate a massive diversity of lymphocytes, each with a unique receptor created by rearranging its DNA. Antigens then select the matching lymphocyte, triggering it to multiply and mount a specific attack. That's clonal selection.
You've got it. That's the fundamental basis of adaptive immune specificity.
Okay, we've covered recognition. Now let's talk more about the invaders themselves. The pathogens, organisms that cause disease, and the process, pathogenesis.
Right, and they come in all shapes and sizes. We generally group human pathogens into 4 major categories based on their characteristics: viruses, bacteria, fungi, and parasites.
And the list within those categories is huge, isn't it? Viruses like polio, HIV, rotavirus, bacteria causing TB, whooping cough, cholera, fungi like candida causing thrush, or tinea causing ringworm, parasites responsible for malaria or filariasis. It's a jungle out there.
It really is. And the immune system needs strategies to deal with all of them. Importantly, where the immune response happens matters a lot. The context is key.
What do you mean by context?
Well, some parts of the body are kind of immunologically privileged, like the brain or the eye. A strong inflammatory response there could cause more harm than the pathogen itself, so immune activity is tightly controlled.
Makes sense. Avoid friendly fire damage.
Exactly. And think about the gut. We have trillions of commensal microbes living there, helping us. The immune system tolerates them in the gut. But if those same microbes get into the bloodstream, big trouble. The immune system reacts very aggressively. Location and context dictate the response.
Before any of that happens, though, the pathogen has to get past our first defenses, right? The physical barriers.
Absolutely critical point. The complex immune pathways we're discussing only really kick in after an organism breaches those initial barriers.
Like our skin.
Skin is the big one, but also mucous membranes lining our airways, gut, reproductive tract. Then there's the acidity stomach acid, the vaginal environment, even sweat is slightly acidic. And cells at these surfaces secrete antimicrobial proteins. It's a multi-layered defense.
And when these barriers fail, That's when infections happen.
Often, yes. Think about how infections start. An animal bite introduces rabies or tetanus past the skin. An insect bite delivers malaria or Lyme disease. Burns are a terrible example. Losing that skin barrier leaves patients incredibly vulnerable to infections. These barriers are unsung heroes. Hashtag 3.2, tailoring the immune response to the assault.
So given this huge variety of pathogens and how they might get in, the immune response needs to be tailored, doesn't it? A one-size-fits-all approach wouldn't work.
Absolutely not. An effective defense has to be specifically designed to counter the nature of the attack. Is the pathogen hiding inside our cells, or is it living in the spaces between them? What's its structure? The immune system needs to figure this out to deploy the right weapons.
How does it figure that out?
Through that recognition process we talked about, interactions between the pathogen or its products and those recognition molecules on our immune cells. These could be receptors on the cell surface or soluble molecules floating around. The specific molecules that get triggered provide clues about the invader.
And binding triggers a cascade.
Yes, a cascade of events that ultimately leads to the pathogen being labeled and destroyed. This involves both the cellular arm cells that kill infected host cells or engulf pathogens, and the humoral arm, those soluble proteins like antibodies that help orchestrate the destruction.
So the strategy differs a lot depending on the type of pathogen, like viruses versus bacteria.
Huge difference. Viruses, for example, are mostly intracellular parasites. They replicate inside our own cells.
So just targeting the virus particles floating around isn't enough.
Right. You have to identify and deal with the infected host cells. This is where specialized T cells, called cytotoxic T lymphocytes, or CTLs, are crucial. They're part of cellular immunity.
How do they find the infected cells?
They can recognize viral proteins displayed on the surface of the infected cell. It's like the infected cell is putting up a little flag saying, "Help, I'm compromised." CTLs see that flag and can then kill the infected cell.
Sacrificing the host cell to stop the virus from spreading.
Often, yes. It's usually necessary for the greater good of the organism, but you can see how problems arise if the virus infects essential immune cells themselves.
Like HIV infecting T helper cells.
Exactly. HIV targets these crucial T helper cells, which normally coordinate the entire immune response, telling B cells what antibodies to make, activating CTLs, and so on. When HIV destroys these helper cells, the whole system starts to collapse. That's what leads to AIDS, the acquired immunodeficiency, making patients vulnerable to all sorts of infections.
Okay, so that's intracellular viruses. What about things living outside cells, like most bacteria or fungi?
For extracellular pathogens, the focus shifts. Recognition molecules on cell surfaces, or soluble ones, like antibodies, patrol the extracellular spaces. B cells and antibodies are major players here. Antibodies can get into nooks and crannies that cells can't, tagging pathogens for destruction.
And what about really big invaders, like parasitic worms? They're too big to be eaten by phagocytes.
Right. The immune system has other tricks for them. Certain immune cells can gather around the parasite and release toxic substances onto its surface. Or the response might trigger expulsion mechanisms, think intense sneezing, coughing, vomiting, or diarrhea, to physically eject the parasite.
So different threats, different tactics. But presumably there's some overlap.
Oh yes, considerable overlap. The pathways interact and support each other, but the key principle is adaptability, tailoring the response to the specific challenge. Hashtag 3.3, how pathogen recognition molecules are encoded and generated.
This tailoring relies on recognition. We talked about specific B and T cell receptors, but you also mentioned an earlier, faster system.
Yes, the innate system's first line of recognition. Pathogens often have common molecular structures that are essential for them but aren't found in us. Think of bacterial cell wall components or unique forms of viral RNA. We call these conserved structures pathogen-associated molecular patterns, or PMPs.
PMPs, like general danger signals shared by groups of microbes.
Kind of, yes. And our innate immune cells have evolved receptors specifically designed to recognize these PMPs. These are the pattern recognition receptors, or PRRs.
So PRRs detect PMPs.
Right, and this recognition is hardwired. The genes for PRRs are encoded directly in our genome, inherited from our parents. They're expressed by many immune cells ready to go. When a PRR binds its PMP, it triggers an immediate response: inflammation, pathogen destruction, and importantly, signals that help shape the next phase of immunity.
So this innate recognition is fast, but maybe less specific. It recognizes general categories of invaders.
Exactly. It's the first alert system. For example, PRRs inside a cell can detect viral components during replication, triggering antiviral defenses in that cell and sending warning signals to neighbors. It allows for rapid containment and also helps the immune system profile the threat early on. Okay, looks like a virus, or this is definitely bacteria.
But pathogens evolve, right? They can change their PMPs or hide them to evade this first line of defense. It's an arms race.
It absolutely is. Pathogens evolve much faster than we do, especially viruses and bacteria with their short generation times and sometimes sloppy replication that introduces mutations. If PRRs were our only defense, pathogens would quickly learn to bypass them.
So how does our immune system keep up? How can it recognize things it's never seen before or things that are constantly changing?
This is where the brilliance of the adaptive immune system comes in, specifically its strategy of generation of diversity. Instead of relying solely on inherited receptors for fixed patterns, the developing B and T lymphocytes employ a strategy that favors randomness in designing their antigen receptors.
Randomness, how does that work?
Each developing B or T cell undergoes a unique process of gene rearrangement. It literally shuffles and edits the DNA segments that code for its antigen receptor, creating a receptor sequence that's essentially random.
So every lymphocyte ends up with a unique receptor generated by chance.
Pretty much, yes. This creates an incredibly diverse pool of lymphocytes, millions upon millions of different specificities. Collectively, this pool has the potential to recognize almost any possible antigen structure, even ones that don't exist yet.
And this happens before encountering the antigen.
Yes. The diversity is generated first in the primary lymphoid organs, bone marrow for B cells, thymus for T cells. It's a high-risk process. Many cells don't make functional receptors or make self-reactive ones and are eliminated. But the survivors circulate, each carrying its unique, randomly generated receptor,
Waiting for its specific antigen to show up and trigger clonal selection.
Exactly. When the antigen appears, it binds only to those few lymphocytes whose random receptors happen to fit. That binding is the signal to activate and proliferate, creating an army of clones specifically tailored to fight that particular invader.
And these randomly generated receptors are not inherited?
Correct. Each individual generates their own unique repertoire. The PRRs of the innate system are inherited. The specific receptors of the adaptive system, B-cell receptors, antibodies, and T-cell receptors, are generated anew in each person. It's Tanagoa's discovery of the DNA recombination mechanism that explained how this was possible.
So 2 layers of recognition. Innate PRRs provide the rapid, general alert, and adaptive, randomly generated receptors provide the highly specific tailored response that follows.
It's a perfect summary.
Okay, but if the adaptive system is generating receptors randomly, isn't there a huge risk that some will accidentally recognize our own body tissues? Like friendly fire on a massive scale.
That is the inherent danger, absolutely. For this system of random generation to work, there must be a mechanism to prevent self-attack. This principle is called tolerance. It's the ability of the immune system to distinguish self from non-self, and specifically not attack self.
Who figured this out?
The concept was really developed by Sir Frank McFarlane Burnett, who theorized that exposure to antigens during development could lead to tolerance. Sir Peter Medawar later provided experimental proof, showing you could induce tolerance in fetal mice by exposing them to foreign cells. They shared the Nobel Prize for this in 1960.
So how does the body enforce tolerance? How does it weed out the self-reactive cells?
It happens primarily during lymphocyte development. As those new B and T cells generate their random receptors in the bone marrow and thymus, they undergo a sort of testing process. They're exposed to a wide range of self-antigens present in those organs.
And if they react strongly to self?
If a lymphocyte's receptor binds too strongly to a self-antigen, it's usually eliminated, essentially ordered to commit suicide, a process called apoptosis. Or sometimes it might be functionally inactivated or repurposed into a regulatory cell that helps suppress other immune responses. This ensures that the lymphocytes released into circulation are generally safe, tolerant of self.
So it's a crucial checkpoint. But there's another model too, right? The danger model.
Yes, proposed by Polly Matzinger. It offers a slightly different perspective. Instead of just focusing on self versus non-self, the danger model suggests the immune system primarily responds to signals of danger or damage.
Danger signals, what are those?
Think about cell death. Normal, programmed cell death happens all the time as tissues renew. That's a good death. Quiet cleanup needed, no alarm bells. But cell death due to injury, infection, or stress, a bad death, often involves the release of specific molecules that aren't normally found outside cells or cellular stress signals. These act as alarmins or danger signals.
And these alarmins trigger the immune response.
The idea is that alarmins, detected by PRRs and other sensors, are the primary trigger for immune activation. So the immune system might encounter a foreign substance, non-self, but ignore it if there are no danger signals. Conversely, it might react to a self-component if it's presented in the context of danger signals, potentially leading to a break in tolerance.
That makes sense. It adds a layer of context. How something is encountered matters as much as what is encountered.
Exactly, and there's quite a bit of evidence supporting it. For example, we know that certain infections can sometimes precede the onset of autoimmune diseases, possibly because the infection provides those danger signals that then trigger reactivity against self-antigens released during the infection.
And failures in tolerance are behind major problems.
Absolutely. On one hand, overly robust self-tolerance can be a problem in cancer, where the immune system might ignore cancerous cells because they still look too much like self, even if they're behaving dangerously.
And on the other hand,
A breakdown in tolerance where the system does attack self is the fundamental cause of autoimmune diseases.
so tolerance is this critical mechanism to prevent self-attack, primarily by eliminating self-reactive lymphocytes during development. And the danger model adds that signals of tissue damage or stress are also key triggers for immune activation.
You've got it.
Okay, we've touched on this already, but let's really clarify the two main arms of the immune system, innate and adaptive. They're distinct but work together, right?
Deeply interconnected, yes. Think of innate immunity as the first responders. It's evolutionarily older, relies on those built-in mechanisms we discussed, physical barriers, chemical barriers, those inherited PRRs that recognize common microbial patterns, PMPs, and pre-existing proteins like complement.
Its job is immediate defense.
Right. Prevent infection in the first place or eliminate common invaders very quickly. It's rapid acting within minutes to hours. But it's not very specific. It recognizes broad categories of threats, and crucially, it doesn't have memory. It responds the same way every time it sees the same pathogen.
Quick and dirty, but no learning.
Pretty much. Then you have adaptive immunity. This is the specialist force. It's much more attuned to subtle differences between antigens, relying on those B and T lymphocytes with their unique, randomly generated receptors.
But it takes longer to get going.
Yes, typically 5 to 6 days for a primary response, partly because you need to find those rare lymphocytes with the right receptor, and partly because it needs signals and information gathered by the innate system first.
So the innate response kind of sets the stage for the adaptive response.
Exactly. But once the adaptive response kicks in, with clonal selection and proliferation, it becomes incredibly powerful and highly specific. It adapts in real time, refining its attack. And, of course, it generates that all-important immunologic memory.
Which is the basis for vaccines.
Precisely. Vaccines aim to generate a strong, long-lasting adaptive memory response.
And adaptive immunity absolutely relies on the innate system.
Profoundly so. The innate response doesn't just fight, it also sends signals, cytokines, and other molecules that activate and direct the adaptive response. It tells the T and B cells what kind of threat they're facing and helps shape the most effective strategy.
This connection was recognized with a Nobel Prize, wasn't it?
Yes, the 2011 Nobel Prize honored discoveries about both innate activation, Butler and Hoffman, and how innate cells like dendritic cells activate the adaptive response, Steinman. It highlighted just how crucial that bridge between the two systems is.
So innate makes first contact, gathers intel, calls for backup, and guides the specialists.
That's a great analogy. Adaptive immunity provides the second, more powerful, highly tailored line of defense, informed by what the innate system encountered. Although sometimes the innate system handles minor incursions all on its own.
Like dealing with a splinter.
Exactly. Local inflammation, phagocytes clean it up, no need to call in the heavy artillery of the adaptive system.
So, summing up the key differences again. Innate is fast, minute hours. Adaptive is slow, days hours. Innate has limited fixed specificity, PRRs. Adaptive has high adaptable specificity, BT cell receptors. Innate has no memory. Adaptive has strong memory. Innate uses barriers, phagocytes, PRRs. Adaptive uses T cells, B cells, antibodies.
Perfect summary. And they collaborate constantly.
Now, unlike, say, the digestive system, which is mostly localized, the immune system has to be everywhere, right? Because pathogens could invade anywhere.
That's a key feature. It has to be highly dispersed. You have immune cells patrolling virtually every tissue, Specialized depots of immune activity like lymph nodes in the spleen and cells circulating constantly in the blood and lymph.
Sentinel cells standing guard everywhere.
Exactly. Tissue-resident immune cells act as local alarms and first responders, kicking off that innate response immediately when a barrier is breached.
But how do the right adaptive cells, those rare B and T cells specific for the current invader, find the action? It seems like finding a needle in a haystack.
That's the place and time problem. The solution involves specialized structures, primarily the lymph nodes. Think of them as communication hubs or meeting points.
How they work?
Fluid from your tissues, called lymph, constantly drains toward lymph nodes, carrying with it any pathogens or antigens picked up locally. At the same time, circulating B and T lymphocytes continuously migrate through the lymph nodes, scanning for their specific antigen.
So the lymph nodes concentrate the antigen and the lymphocytes together.
Precisely. It dramatically increases the chances of that rare, specific lymphocyte encountering its antigen. Lymph nodes provide the perfect microenvironment for that initial activation, clonal selection, and the start of the adaptive immune response.
But with cells scattered everywhere and meeting in lymph nodes, how do they coordinate everything? How do they talk to each other?
Communication is critical. It happens through direct cell-to-cell contact, but also, very importantly, through secreted messenger proteins called cytokines.
Cytokines. We hear that term a lot.
They're essential signaling molecules. One cell releases a cytokine, it binds to a specific receptor on another cell, and that triggers changes inside the target cell. Maybe it gets activated, starts dividing, or changes its function.
And there are different types of cytokines.
Many different types, with different effects. A specific subset called chemokines act like chemical trails, guiding immune cells to migrate towards the site of infection or inflammation. They're crucial for recruitment.
So cytokines and chemokines orchestrate the inflammatory response we feel. The swelling, heat, redness.
They're major drivers of it, yes. They recruit immune cells and cause blood vessels to become leaky, allowing fluid and more cells to enter the tissue to fight the infection. It's all part of a coordinated process.
Can you walk us through a typical sequence of events, like from the moment a pathogen gets in?
Sure. Imagine a cut in your skin. 1. Pathogen gets past the skin barrier. 2. Resident innate cells, like macrophages, detect it using their PRRs. They start engulfing pathogens and releasing cytokines and chemokines, the alarm bells. Inflammation begins locally. 3. Some free pathogens and some of the innate cells that have eaten pathogens travel via lymphatic vessels to the nearest lymph node. They're carrying the antigen intel. 4. Meanwhile, naive B and T lymphocytes are circulating through that lymph node anyway, arriving via the bloodstream. 5. Inside the lymph node, a T cell or B cell whose receptor happens to match the antigen being presented gets activated. Clonal selection happens. It starts dividing rapidly, making many copies. This is the adaptive response kicking off. These cells differentiate into effector cells, ready to fight. B cells start producing specific antibodies. 6. These newly activated effector T cells, B cells, now often plasma cells making antibodies, and the antibodies themselves leave the lymph node and enter the bloodstream. 7. They circulate around the body, but they're drawn to the original site of infection because of the ongoing inflammation signals, chemokines. They exit the blood vessels there. 8. Finally, they arrive at the infection site, find the pathogens, often tagged by antibodies or being handled by innate cells, and launch their specific attack. T cells might kill infected cells, antibodies neutralize toxins, or tag bacteria for destruction. This is the effector phase, clearing the infection.
And after the battle is won.
Many of the effector cells die off, but a crucial population of memory B and T cells remains behind long term. They remember the specific antigen and provide that rapid, enhanced response if the same pathogen ever returns.
It's an incredibly intricate, coordinated dance between innate and adaptive, local and systemic responses.
It truly is. Dispersed components, communication hubs, signaling molecules, all working together. Hashtag TAG 4.3, the power of adaptive memory.
Let's focus on that memory aspect. You said it's unique to the adaptive system.
Yes, immunologic memory is a hallmark of adaptive immunity. It's the ability to mount a faster, stronger, more effective response upon subsequent encounters with the same pathogen. The immune system literally learns and remembers.
The first time you encounter a new pathogen, that's the primary response.
You're right. The primary response is where those naive lymphocytes get selected, activated, proliferate, and differentiate, incorporating signals from the innate system to tailor the attack. It takes time, maybe a week or so to peak.
But then, if you meet that same pathogen again later,
That triggers a secondary response. And this is where the memory cells shine. These are descendants of the cells activated during the primary response. They're already primed, more numerous, and react much more quickly and vigorously.
So the secondary response is faster and stronger.
Much faster, much stronger, often peaks higher and lasts longer. The memory cells basically pick up where the primary response left off, but better and faster.
And this memory can last a long time.
Decades, even a lifetime for some infections or vaccines. That's why you often don't get sick from the same virus twice, or why vaccines provide long-term protection. The secondary response often wipes out the pathogen before you even feel symptoms.
So memory cells save the strategy, not the pathogen itself.
Exactly. They retain the blueprint for the effective response developed during the primary encounter. That's the foundation of vaccination, safely inducing that primary response and creating memory.
What about booster shots? Why do some vaccines need them?
Sometimes a single exposure isn't enough to generate a strong enough or long-lasting enough memory response for full protection. Booster shots are essentially planned secondary exposures to the antigen. Each booster triggers another round of adaptive response, further expanding the pool of memory cells and refining their effectiveness, ensuring protection is robust and durable.
So the primary response creates the memory cells, and the secondary response, triggered by re-exposure or boosters, utilizes those memory cells for a much faster, more powerful defense. It's biological learning in action.
Perfectly stated.
Okay, we've talked a lot about how amazing the immune system is when it works right, but sometimes it doesn't. It can malfunction.
Yes, absolutely. While it's usually protective, failures or dysregulations do happen. They might be rare overall, but they can be dramatic and cause significant disease.
And sometimes we ask the immune system to deal with things it didn't really evolve for, like transplants or cancer.
Exactly. Those pose unique challenges. Broadly speaking, immune dysfunction happens when the regulation goes wrong. It either attacks things it shouldn't or fails to attack things it should. We can group these problems into roughly 4 categories.
Who are they?
First, hypersensitivity, like allergies. That's an overreaction, an overly aggressive attack on harmless foreign things. Second, autoimmune disease, where the system mistakenly attacks the body's own tissues.
Okay, attacking it appropriately. What else?
Third, immune deficiency, where the response is too weak or insufficient to fight off infections properly. And fourth, something we're increasingly recognizing, immune imbalance, a kind of dysregulation leading to excessive inflammation or problems with the off switches. Hashtag 5.2 hypersensitivity reactions, allergies, and anaphylaxis.
Let's start with hypersensitivity.
Yeah.
Allergies and asthma are common examples.
Classic examples. They result from inappropriate, over-the-top immune responses to common environmental substances that are actually harmless things like pollen, dust mites, certain foods, pet dander. We call these substances allergens.
It's like the immune system is being way too sensitive. Where did this idea come from?
The realization that the immune system could cause harm, not just protection, came around 1902 from Charles Richet.
What did he find?
He was trying to immunize dogs against jellyfish toxins. He gave them a small dose, expecting it to protect them. But when he gave them a second, even tiny dose later, they had an immediate, severe, and often fatal reaction.
The opposite of protection.
Exactly. He called it anaphylaxis anti-protection. It was this extreme, rapid overreaction. Richet won the Nobel Prize for this discovery in 1913. We still use the term anaphylaxis for severe, life-threatening allergic reactions today.
Thankfully, most allergic reactions in humans aren't that severe, right?
Correct. While some can be T-cell mediated, the most common types, like hay fever or food allergies, typically involve a specific type of antibody called immunoglobulin E, or IgE.
IgE, what does it do?
IgE antibodies bind to allergens. When this happens on the surface of certain immune cells, like mast cells, it triggers those cells to release a flood of chemicals. Histamine is a famous one that causes irritation and inflammation.
Leading to the symptoms we associate with allergies.
Yes. Sneezing, runny nose, itchy eyes from hay fever, wheezing and difficulty breathing in asthma, skin reactions like hives or eczema. And in severe cases, anaphylaxis can cause airway constriction, a drop in blood pressure.
Yeah.
It's a medical emergency. Allergies are a huge health burden worldwide.
Which brings us to that interesting idea, the hygiene hypothesis. Why are allergies and asthma seemingly increasing in industrialized countries?
It's a striking trend. Asthma rates, for example, roughly doubled in many developed nations over about 25 years. The hygiene hypothesis proposes a link between this rise and our modern, cleaner lifestyles.
Cleaner lifestyles are bad? That seems counterintuitive.
The idea isn't that hygiene itself is bad, but that reduced exposure to a wide range of microbes, especially early in life, might be preventing our immune systems from developing properly balanced regulatory mechanisms. Widespread antibiotic use, smaller families, less exposure to farm environments, cleaner water, and food, all these might contribute.
So less exposure to dirt and germs means the immune system doesn't learn to regulate itself properly.
That's the core idea. It was first proposed by Strachan in '89, linking hay fever and family size. Now it's expanded to include asthma, other allergies, even some autoimmune conditions like inflammatory bowel disease.
Is there evidence for this?
Quite a bit, mostly correlational. Studies show that kids who grow up on farms, have older siblings, attend daycare early, or live in less sanitized environments often have lower rates of allergies and asthma. Exposure seems key.
Exposure to what specifically?
It seems exposure to certain bacteria and maybe even parasites like helminths, worms, might be particularly important for training the immune system's regulatory side. Not so much viruses. People are even experimenting with controlled helminth exposure as therapy for some conditions.
So the theory is that our immune systems co-evolved with constant microbial exposure.
Yeah.
These old friends, both good and bad microbes, engaged our innate PRRs and helped tune the adaptive system to find a balance between attack and tolerance, aggression and inhibition.
Exactly. Without that constant tuning early in life, the hypothesis suggests the immune system might become biased towards overreaction, towards inflammation.
Animal studies support this too.
Yes. Mice raised in sterile, pathogen-free environments are often much more susceptible to developing autoimmune diseases like type 1 diabetes compared to mice living in dirtier conditions. It strongly suggests environment plays a huge role, alongside genetic predisposition. Allergies do tend to run in families.
So maybe Grandma was right about letting kids play in the dirt. It's good for their immune system.
There might be more truth to that than we realized. Hashtag tagged 5.3 autoimmune disease.
Okay, so hypersensitivity is an overreaction to something harmless outside the body. What happens when that misdirected attack turns inward against the body itself? That's autoimmunity.
Precisely. Autoimmune disease occurs when there's a breakdown in self-tolerance. The immune system fails to distinguish self from non-self, or maybe it misinterprets a self component as dangerous due to those danger signals we talked about.
And the result is friendly fire.
Yes, the immune system launches an attack against the body's own tissues or organs. This leads to chronic inflammation and damage, causing a wide range of debilitating diseases.
And the specific disease depends on what tissue is being attacked.
Exactly. If the immune system attacks the myelin sheath around nerves, you get multiple sclerosis, causing neurological problems. If it attacks the lining of the intestines, you get Crohn's disease or ulcerative colitis. If it attacks the joints, you get rheumatoid arthritis.
What causes this breakdown in tolerance?
It's complex and still not fully understood. It likely involves a combination of genetic susceptibility. Certain genes make you more prone to environmental triggers. Things like infections, maybe exposure to certain chemicals, potentially even factors related to the microbiome or diet, could play a role in tipping someone with a genetic predisposition over the edge into autoimmunity. It's a major area of research. Hashtag 5.4 immune deficiency.
Then there's the flip side, not enough immune response. Immune deficiency.
Right. Immune deficiency means some component of the innate or adaptive system is missing or defective. This can range from relatively minor issues to being completely life-threatening.
And it can be inherited or acquired.
Correct. Primary immunodeficiencies are genetic inherited conditions. Some are quite common but mild, like selective IgA deficiency, where people lack one type of antibody. They might get more frequent respiratory or gut infections or sometimes notice nothing.
But some primary deficiencies are very severe.
Yes. The most extreme example is severe combined immunodeficiency, SCID, sometimes called bubble boy disease. These individuals lack functional B and T cells, essentially having no adaptive immunity. Without treatment, like a bone marrow transplant, they usually die from common infections very early in life.
And secondary immunodeficiencies are acquired later.
Yes, caused by external factors that damage or disrupt the immune system. And tragically, the single most common cause worldwide is severe malnutrition.
Malnutrition, more common than anything else.
By far. Lack of sufficient protein, calories, or essential micronutrients severely compromises immune function. It affects hundreds of millions, maybe billions of people. It's why common childhood diseases like pneumonia, diarrhea, or malaria are so much deadlier in malnourished populations, especially children. Tackling malnutrition would probably do more than almost anything else to reduce global deaths from infectious diseases.
That's a sobering thought. What's another major cause of secondary immunodeficiency?
The most well-known is probably acquired immune deficiency syndrome, AIDS, caused by the human immunodeficiency virus, HIV.
How does HIV cause deficiency?
As we mentioned, HIV specifically infects and destroys those critical T helper cells. Our immune system struggles to clear the virus completely. Over time, the loss of T helper cells cripples the ability to coordinate immune responses, leading to the profound immunodeficiency of AIDS.
Although treatments have improved outcomes dramatically.
Immensely. Anti-HIV drugs can keep the virus suppressed and allow the immune system to recover significantly, turning HIV into a manageable chronic condition for many. But access to these drugs is still a huge issue in many parts of the world hardest hit by the epidemic.
And a key sign of any severe immunodeficiency is opportunistic infections.
Yes, opportunistic infections are caused by microbes that are usually harmless to people with healthy immune systems, but can cause severe disease when immunity is weakened. The fungus Candida albicans is a classic example.
Candida, that causes thrush.
Exactly. In immunocompetent people, it usually lives harmlessly on skin and mucous membranes. But in someone with AIDS or SEID or undergoing intense chemotherapy, Candida can overgrow, causing thrush in the mouth or vagina. If it spreads systemically, it can be life-threatening. Seeing unusual opportunistic infections like this was actually one of the first clues that led to the recognition of AIDS as a new disease back in the early 80s. Hashtag 5.5 immune imbalance.
You also mentioned a fourth category, immune imbalance. This sounds subtler than just too much or too little response.
It is. We often focus on the attack functions of the immune system, but it's equally important to have effective braking or inhibitory mechanisms. A healthy immune response requires a delicate balance between activating go signals and inhibitory stop signals. It's like driving a car, you need both the gas pedal and the brake.
So immune imbalance is when the brakes fail or the gas pedal gets stuck.
That's a good analogy. Problems with the inhibitory side can be just as damaging as problems with the attack side. And there's growing evidence linking chronic, uncontrolled inflammation, essentially, the gas pedal being stuck onto a huge range of non-infectious chronic diseases.
We already talked about allergies and autoimmunity.
Yes, those are clear examples of immune overactivation. But the links now extend to conditions like cardiovascular disease, type 2 diabetes, obesity.
Really? Heart disease and diabetes linked to immune imbalance.
Yes, chronic low-grade inflammation seems to play a significant role in the development of atherosclerosis, hardening of the arteries, and insulin resistance. And even more recently, researchers are finding connections between immune dysregulation, inflammation, and even neurological or psychiatric conditions like depression, bipolar disorder, maybe even autism spectrum disorders.
Wow, so what could be throwing this balance off? What's weakening the brakes or flooring the gas?
Several factors are prime suspects, and they likely interact. The microbiome, the community of microbes living in and on us, especially in the gut, seems critical. Our modern Western diet is another major factor, and chronic stress also clearly impacts immune function.
How do they interact?
It's complex and bidirectional. Changes in diet affect the microbiome. The microbiome influences immune development and regulation. Stress hormones affect immune cells. Immune signals can affect the brain and metabolism. There's mounting evidence that changes in our gut microbes, perhaps driven by diet and lifestyle, might be leading to a loss of important regulatory signals, a paucity of brakes, leaving the inflammatory gas pedal pressed down more often.
This state of microbiome disruption is called dysbiosis.
Yes, dysbiosis. It's thought to contribute to immune imbalance, chronic inflammation, increased gut permeability, leaky gut, which then potentially impacts metabolic health, neurological function, and contributes to all those chronic diseases we mentioned, type 2 diabetes, inflammatory bowel disease, maybe even mood disorders. It highlights how deeply interconnected everything is.
Let's switch gears slightly to transplantation. Here, the immune system doing its job correctly is actually the problem, isn't it?
Exactly. The immune system is designed to recognize and attack foreign cells. A transplanted organ from another person is foreign tissue. So the immune rejection of a transplant isn't a failure of the immune system. It's the system working precisely as it evolved to recognizing non-self and responding to the danger signals inevitably caused by the surgery itself.
Which makes transplantation incredibly difficult.
It does. We need to give powerful immunosuppressive drugs to prevent rejection. But the downside is that these drugs dampen the entire immune system, leaving the patient vulnerable to infections and even some cancers. It's a constant balancing act.
Has research into overcoming rejection taught us a lot about immunology?
Immensely. Trying to solve the transplant problem has driven major discoveries. Carl Landsteiner figuring out ABO blood groups was essential for safe blood transfusions, a form of transplant. Discovering the major histocompatibility complex, MHC genes, by Snell, Dossett, and Benacerraf, another Nobel Prize, identified the key molecules that trigger rejection between genetically different individuals. And pioneers like Thomas and Murray developed the techniques that made organ transplants clinically feasible.
So the big goal now is inducing tolerance specifically to the transplant.
That's the holy grail.
Sure.
Tricking the immune system into accepting the foreign organ as self without shutting down its ability to fight off infections. We're making progress, but it's still a major challenge.
Finally, let's talk about cancer. You mentioned earlier that sometimes self-tolerance works too well, and the immune system ignores cancer cells because they arise from self.
Yes, it's another complex situation. Cancer is essentially our own cells growing uncontrollably. Because they originate as self, the mechanisms of self-tolerance can prevent the immune system from recognizing them as dangerous, even when they clearly are.
But don't cancer cells sometimes look different? Don't they have mutations or express weird proteins?
They often do. Many tumors express unique antigens, tumor-specific antigens, or re-express proteins normally only found during fetal development, tumor-associated antigens. These could theoretically be targets for the immune system.
So why doesn't the immune system always eliminate them?
Several reasons. Sometimes the antigens aren't different enough, or they aren't presented effectively. Tolerance mechanisms might still suppress the response, and cancer cells themselves are masters of evasion. Because they divide rapidly and have genetic instability, they can quickly evolve ways to hide from immune detection, suppress local immune responses, or even kill incoming immune cells.
So the immune system does try to fight cancer. It's not just ignoring it.
Oh, absolutely. There's a constant interplay called cancer immunosurveillance. The immune system does recognize and eliminate many precancerous or cancerous cells. We know this partly because people with weakened immune systems, like transplant recipients on immunosuppressants or people with AIDS, have significantly higher rates of certain cancers.
But the relationship is complicated. Inflammation can sometimes help cancer grow.
Yes, it's a double-edged sword. Chronic inflammation can actually promote tumor development in some cases, and the immune system can exert pressure on the tumor, selecting for more aggressive, evasive cancer cells over time. So the immune response can be both protective and contribute to disease progression.
Which makes cancer immunotherapy, harnessing the immune system to fight cancer, so exciting, but also challenging.
Exactly. It's one of the most promising areas of cancer research, designing therapies that boost the anti-tumor functions of the immune system while blocking the pathways that help the tumor evade or suppress immunity. That's the goal. Checkpoint inhibitors are a prime example of this success.
It sounds like our understanding has come incredibly far.
It really has, especially in the last few decades. But there's still so much to learn about the immune system's intricacies and its connections to overall health. This deeper knowledge holds immense promise for developing innovative treatments for cancer, autoimmune diseases, allergies, and maybe even for preventing chronic conditions linked to immune imbalance. And of course, for being better prepared for the next emerging infectious threat. #tag outro.
Wow. What an incredible journey through the immune system today. We've gone from those first ancient observations of immunity through the breakthroughs of Jenner and Pasteur with vaccines, right up to the incredibly complex dance between innate and adaptive immunity, clonal selection, tolerance. It's just amazing.
It really is, and we've seen how it recognizes threats, tailors its response, remembers its enemies, but also how it can go wrong. Hypersensitivity, autoimmunity, immunodeficiency, and the challenges posed by transplants and cancer.
It really drives home that idea of the immune system constantly walking this tightrope, doesn't it? Balancing aggression against regulation, attack against tolerance, and how sensitive it is to our environment, our microbes, our lifestyle.
Absolutely. And the pace of discovery is just accelerating. It feels like we're constantly uncovering new layers of complexity and new connections, opening doors to treatments and asking questions we couldn't have even imagined just a decade or two ago. It's a thrilling field.
So thinking about all this, What does it mean for you listening right now, given this intricate balance, these deep links to your gut microbiome, your diet, even your stress? Are there proactive things you might think about to support your own sort of immune ecological balance?
That's the provocative thought, isn't it? Could understanding and maybe even nurturing our body's microbial communities be the next big frontier in personalizing health? Moving beyond just avoiding pathogens to actively cultivating a state of internal harmony, it really extends the whole idea of hygiene, perhaps, to something much broader. Definitely something to ponder.